Well, I thought there'd be a podium, so I'm a bit scared. Chris asked me to tell again how we found the structure of DNA. And since, I follow his orders, I'll do it. But it slightly bores me. And I wrote a book. So I'll say something. I'll say a little about, how the discovery was made and why Francis and I found it. And then I hope maybe I have at least 5 minutes to say what makes me tick now. Back of me is a picture of me when I was 17. I was at the University of Chicago in my third year. And I was in my third year because the University of Chicago let you in after 2 years of high school, so it was fun to get away from high school, and because I'm very small, no good in sports or anything like that. But I should say that my background, my father was raised to be an Episcopalian and Republican, but after 1 year of college he became an atheist and a Democrat, and. My mother was Irish Catholic, but she didn't take religion too seriously, and by the age of 11 I was no longer going to Sunday Mass and going on birdwatching walks with my father. So early on I heard of Charles Darwin. I guess, he was the big hero, and, you understand life as it now exists through evolution. And at the University of Chicago, I was a zoology major, and thought I would end up, if I was bright enough, maybe getting a PhD from Cornell in ornithology. Then, in the Chicago paper, there was a review of a book that, called What is Life by the great physicist Schrödinger. And that, of course, had been a question I wanted to know. Darwin explained life after it got started, but what was the essence of life? And Schrödinger said the essence was information, present on our chromosomes, and it had to be present on a molecule. I never really thought of molecules before, chromosomes, but this was a molecule, and somehow the information was probably present in some digital form. And there was a big question, how did you copy the information? So that was the book, and so from that moment on, I wanted to be a geneticist, understand the gene, and through that understand life. So I had, a hero at a distance, wasn't a baseball player, it was Linus Pauling. And so I applied to Caltech, and they turned me down. So I went to Indiana, which was actually as good as Caltech in genetics, and besides they had a really good basketball team. So I had a really quite happy life at Indiana, and it was in Indiana I got the impression that the gene was likely to be DNA, and so when I got my PhD I should go in search of DNA. So I first went to Copenhagen because I thought, well, maybe I could become a biochemist, but I discovered biochemistry was very boring. It wasn't going anywhere toward, saying what the gene was. It was just nucleosides. And, that's the book, little book. You can read it in about 2 hours. And, but then I went to a meeting in Italy, and there was an unexpected speaker. He wasn't on the program, and he talked about DNA. This was Morris Wilkins. He was trained as a physicist, and after the war he wanted to do biophysics, and he picked DNA because DNA had been shown at the Rockefeller Institute to possibly be the genetic molecules on the chromosomes. Most people believed it was proteins. But Wilkins, thought DNA was the best bet, and he showed this X-ray photograph. And it was sort of crystalline. So DNA had a structure, even though it, all probably different molecules carried different set of instructions. So there was something universal about the DNA molecule. So I wanted to work with him, but he didn't want a former bird watcher. And I ended up in Cambridge, England. So I went to Cambridge because It was really the best place in the world then for X-ray crystallography. And X-ray crystallography is now subject to, chemistry departments. But in those days, it was in the domain of the physicists. So the best place for X-ray crystallography was in the Cavendish laboratory at Cambridge. And there I met Francis Crick. I went there without knowing him. He was 35, I was 23. And within a day, we decided that maybe we could take a shortcut to finding the structure of DNA, not solve it by a rigorous fashion, but build a model, a molecular model using some coordinates, some length, all that sort of stuff from X-ray photographs, but just ask what the molecule, how should it fold up? And the reason for doing so is the center of the photograph is Linus Pauling. About 6 months before, he proposed the alpha helical structure for proteins. And in doing so, he banished the man on the right, Sir Lawrence Bragg, who was the Cavendish professor. This is photographed several years later when Bragg had caused a smile. He certainly wasn't smiling when I got there because he was somewhat humiliated by Pauling getting the alpha helix and the Cambridge people failing because they weren't chemists. And certainly neither Crick or I were chemists. So we tried to build a model, and Francis knew Wilkins. Wilkins said he thought it was the helix. X-ray diagram he thought was compatible with the helix. So we built a 3-stranded model. The people from London came up, Wilkins and this collaborator or possible collaborator, Rosalind Franklin, came up and sort of laughed at our model. They said it was lousy, and it was. So we were told to build no more models. We were incompetent. And so we didn't build any models, and Francis sort of continued to work on proteins, and basically I did nothing. Except read. Basically reading is a good thing. You get facts. And we kept telling the people in London that Linus Pauling is going to move on to DNA. If DNA is that important, Linus will know it, he'll build a model, and everyone will be scooped. And in fact, he had written the people in London, could he see their X-ray photograph? And they had the wisdom to say no, so he didn't have it. But there was ones in the literature. Actually, Linus didn't look at them that carefully. But about 15 months after I got to Cambridge, or rumors began to appear from Linus Pauling's son, who was in Cambridge, that his father was now working on DNA. And so one day Peter came in, as Peter Pauling, and gave me a copy of his father's manuscripts. And boy, I was scared, because I thought, we may be scooped. I have nothing to do, no qualifications for anything. So there was the paper, and he proposed a 3-stranded structure, and I read it, and it was crap. So this was, unexpected from the world. And so it was held together by hydrogen bonds between phosphate groups. Well, at the peak pH that cells have, around 7, those hydrogen bonds couldn't exist. We rushed over to the chemistry department and said, could Pauling be right? And Alex Todd said no, so we were happy. And we were still in the game, but we were frightened that someone at Caltech would tell Linus that he was wrong. And so Bragg said build models, and a month after we got the Pauling manuscript, I should say, I took the manuscript to London to show the people, that Linus was wrong and they were still in the game. And they should immediately start building models, but Wilkins said no. Rosalind Franklin was leaving in about 2 months, and after she left, he would start building models. And so I came back with that news to Cambridge, and Bragg said, build models. Well, of course, I wanted to build models. And there was a picture of Rosalind. She really, in one sense, she was a chemist, but really she had been trained, she didn't know any organic chemistry or quantum chemistry. She was a crystallographer. And I think part of the reason she didn't want to build models is she wasn't a chemist, whereas Pauling was a chemist. And so Crick and I, started building models, and I learned a little chemistry, but not enough. Well, we got the answer on 28th of February, '53, And it was because of a rule, which to me is a very good rule: never be the brightest person in the room. And we weren't. I mean, we weren't the best chemists in the room. I went in and showed them a pairing I'd done, and Jerry Donahue, he was a chemist. He said, it's wrong. You got the hydrogen atoms are in the wrong place. I just put them down like they were in the books. He said they were wrong. So the next day, you know, after I thought, well, he might be right, so I changed locations, and then we found the base pairing, and Francis immediately said the chains run in opposite directions, and we knew we were right. So it was a pretty, you know, it all happened in about 2 hours. You know, from nothing to big. And we knew it was big because, you know, if you just put A next to T and G next to C, you have a copying mechanism. So we saw how genetic information is carried. It's the order of the 4 bases, so in a sense it is a sort of digital type information. And you copy it by going from strand separating. So it's, you know, if it didn't work this way, you know, you might as well believe it because you didn't have any other scheme. But that's not the way most scientists think. Most scientists are really rather dull. They say we won't think about it until we know it's right. But, you know, we thought it was at least 95% right or 99% right, so think about it. The next 5 years, there were essentially something like 5 references to our work in Nature. None. And so we were left by ourselves and trying to do the last part of the trio. What does the genetic information do? And it was pretty obvious that it provided the information to an RNA molecule, and then how do you go from RNA to protein? For about 3 years, I tried to solve the structure of RNA. It didn't yield, didn't give good X-ray photographs. I was largely unhappy. A girl didn't marry me. It was really, you know, sort of a shitty time. So there's a picture of Francis and I before I met the girl, so I'm still looking happy. But there is what we did when we didn't know where to go forward. We formed a club. And called it the RNA Tie Club. George Gamow, the great physicist, he designed the tie. He was one of the members, and the question was, how do you go from a 4-letter code to the 20-letter code of proteins? Feynman was a member, and Teller, and friends of Gamow. But that's the only photo. No, we were only photographed twice, and in both occasions, you know, one of us was missing the tie. There's Francis up on the upper right, and Alex Rich, the MD turned crystallographer, is next to me. This was taken in Cambridge in September of 1955. And I'm smiling, sort of forced, I think, because the girl, boy, she is gone. And so I didn't really get happy until 1960, because then we found out basically, you know, that there are 3 forms of RNA, and we knew basically DNA provides the information, RNA provides the information, protein, and that led Marshall Nierenberg, you know, to take RNA, synthetic RNA, put it in a system making protein. He made phenylalanine, poly-U made phenylalanine. So that's the first cracking of the genetic code, and it was all over by 1966. So that's what Chris wanted me to do. It was so what happened since then? Well, at that time, I should go back. When we found the structure of DNA, in my first talk at Cold Spring Harbor, the physicist Leo Szilard, he looked at it and he said, "Are you going to patent this?" But he knew patent law and knew we couldn't patent it because there was no use for it. And so DNA didn't become a useful molecule, and the lawyers didn't enter into the equation until 1973, 20 years later, when Boyer and Cohen in San Francisco and Stanford came up with their method of recombinant DNA, and Stanford patented it and made a lot of money. That is, they patented something which, you know, could you do useful things, and then they learned how to read the letters of the code, and boom, we've had a biotech industry, and but we're still a long ways from answering a question which sort of dominated my childhood, which is how do you nature nurture? And so I'll go on. I'm already out of time, but this is Michael Wigler, a very, very clever mathematician turned physicist, and he developed a technique which essentially will let us look at sample DNA at eventually 1 million spots along it. There's a chip there, a conventional one, then there's one made by photolithography by a company in Madison called Nimblegen, which is way ahead of Affymetrix, and we use their technique. And what you can do is sort of compare DNA of normal, say, versus cancer, and you can see on the top that cancers which are bad show insertions or deletions. So the DNA is really badly mucked up, whereas if you have a chance of surviving, the DNA isn't so mucked up. So we think that this will eventually lead to what we call DNA biopsies. Before you get treated for cancer, you should really look at this technique and get a feeling of the face of the enemy. It's only a partial look, but I think it's going to be very, very useful. So we started with breast cancer because there's lots of money for it, no government money. And now I have a sort of vested interest. I want to do it for prostate cancer. So, you aren't treated if, it's not dangerous. And but Wigler, besides looking at cancer cells, looked at normal cells and made a really sort of surprising observation, which is all of us have about 10 places in our genome where we've lost a gene or gained another one. So we're sort of all imperfect. And the question is, well, if we're around here, these losses or gains might not be too bad. But if these deletions or amplifications occurred in the wrong gene, maybe you really are sick. So the first disease he looked at was autism. And the reason that we looked at autism was we had the money to do it. To look at an individual was about $3,000, and the parent of a child with Asperger's disease, the high intelligence autism, had sent his thing to a conventional company. They didn't do it, couldn't do it by conventional genetics, but just scanning it, we began to find genes for autism, and you can see here, there are a lot of them. So a lot of autistic kids are autistic because they just lost a big piece of DNA. I mean, big piece at the molecular level. We saw one autistic kid, 5 million bases just missing from one of his chromosomes. We haven't yet looked at the parents, but the parents probably don't have that loss or they wouldn't be parents. Now, so our autism study is just beginning. We got $3 million. I think it'll cost at least 10 to 20 before you'd be in a position to help parents who've had an autistic child or think they may have an autistic child, and can we spot the difference. So this same technique should probably look at all. It's a wonderful way to find genes. And so I'll conclude by saying we've looked at 20 people with schizophrenia. And we thought we probably had to look at several hundred before we got a picture. But as you can see there, 7 out of 20 had a change which was very high. And in the controls, there were 3. So what's the meaning of the controls? Were they crazy also and we didn't know it, or, were they normal? I would guess they're normal. And what we think in schizophrenia is there are genes that predispose you. And whether this is one that predisposes, and then there's only a subsegment of the population that's capable of being schizophrenic. Now, we don't have really any evidence of it, but I say, to give a hypothesis, the best guess is that if you're left-handed, you're prone to schizophrenia. 30% of schizophrenic people are left-handed, and schizophrenia has a very funny genetics, which means 60% of the people are genetically left-handed, but only half of it show it. I don't have the time to say it. Now, some people who think they're right-handed are genetically left-handed. Okay, I'm just saying that if you think, oh, I don't carry a left-handed gene. So therefore, my children won't be at risk of schizophrenia. You might. Okay. So it's, to me, an extraordinarily exciting time. We ought to be able to find the gene for bipolar. There's a relationship. And if I had enough money, we'd find them all this year. Thank you.